next speaker is Salomon.
Okay, so good afternoon, everyone. First, I want to thank the organizers for the opportunity to present our work here today, because it has nothing to do with COVID. I'm going to talk about how we engineer antibacterial biotreatments. And so you're all aware that we have a problem with antibiotic-resistant bacteria. A recent report determined that in the year 2019, more than 1.2 million deaths worldwide can be directly attributed to antibiotic-resistant bacteria, and many more can be associated with it. And a report that was commissioned by the UK government in 2014 predicted that at the current rate, by the year 2050, antibiotic-resistant bacteria are going to be the #1 cause of death, surpassing cancer and diabetes. While this is happening, the number of antibiotics, classical antibiotics that are being released to the market, diminishes with every decade that passes. So obviously there's a dire need to develop alternative strategies to classical antibiotics to treat this problem. And the scientific community has risen to the challenge, and we have all kinds of strategies that people are working on, especially dealing with bacteriophages, probiotics, antimicrobial peptides, and a few other things. And we need to remember that it's not enough to work on one strategy. If we learn anything from the problems that we have with antibiotics, is that we need a multifaceted approach to stay ahead in the arms race against these bacteria. And this is where we come in. And in the past few years in my lab, we've been studying an antibacterial machine that bacteria use against each other during competition. So these machines are basically these nanotubes that, upon contraction of an outer sheath, shoot out a missile-like structure that is called a tail tube. And this tail tube punctures a neighboring bacterial cell. And because it's decorated with toxic proteins that we call effectors, it can deliver these effectors into a neighboring cell. And these effectors have antibacterial activities against very conserved bacterial components. And so we reasoned that maybe we can steal some ideas from bacteria or even adapt this system as a novel strategy for a biotreatment against drug-resistant bacteria. And to do so, we reasoned that we're going to need to do this in a safe bacterium, something that we can administer to patients or that we can release to the environment without causing any harm. We want the system to be inducible. So this is, first of all, very energetically costly to the bacteria that make it, so we don't want it constitutively active. And we also don't want it to kill everything in its path, because then we're going to cause dysbiosis that we really don't want to have. And we want the system to be customizable. So we want to be able to change different aspects of the system. For example, what are the effectors, these toxins that are going to be delivered, so we can control the toxicity range of the system. We want to be able to control how it's going to be induced. And by physically separating the different components, we can gain also a safety thing that will prevent the pathogens we want to kill from stealing a functional antibacterial system that will actually enhance their own fitness. And the design we came up with is the following. We decided to use a bacterium called Vibrio nitrigens as our host for this platform. Vibrio nitrigens is a gram-negative marine bacterium, and it's considered safe. There are no known virulence determinants, no known antibacterial determinants. It grows under a wide range of conditions, and we can genetically manipulate it. What we had in mind is to put a type 6 secretion system from another bacterium with antibacterial activities into this bacterium and separate from it an on-off switch, physically separate it, to put the on-off switch somewhere on the chromosome of Vibrio nitrigens, and have this on-off switch regulated by responding to some external condition. And so that when the external condition is present, the on-off switch will be expressed, activate the type 6 secretion system, and hopefully effectors that we control their identity to kill a target bacterium. Now this is plasmids and stuff because we're still talking about a proof of concept. This is not the final product. So into this Vibrio nitrigens, we decided to introduce a type 6 secretion system from a pathogen called Vibrio parahemolyticus. So obviously we can't use a pathogen, so we have to take the type 6 secretion system from that. And this system is encoded on a large gene cluster. This is a 50 kilobase cluster. encoding all the necessary components to make a functional system that I showed you, including also antibacterial effectors that will mediate the toxicity and regulators. So our first question was, will this secretion system work in vibrio regions? Can it mediate antibacterial killing? So we cloned this onto a plasmid and put it into vibrio regions. Or we took vibrio regions and put an empty vector as control. And then we did a competition assay where we mixed these bacteria together with some prey bacterium, and we measured the viability of the prey bacterium at the beginning of this competition. That's what you see here in the red triangles. And then after 4 hours of competition, that's what you see in the blue circles. And you see that when the type 6 system was present in vibrio regions, the prey bacteria died. Okay, this is orders of magnitude in the y-axis. So the system can be functional and mediate antibacterial killing in vibrio regions. But we want it to be inducible. So we had to find something that can be an on-off switch. And by doing some epistasis experiments, biochemistry, and genetics, we identified this small gene here, VP1407, as a possible on-off switch. Because when we deleted this gene, all of the system was shut off. None of the operons were expressed. But when we put it back, all of the operons were turned on, including its own operon. So first we deleted this gene to make sure that it really shuts off the system, and did the competition assay again. And you can see that now the system, which we call inducible because we hoped that it can be induced by this on-off switch, no longer can mediate killing of the prey. You see that the prey was growing over time. What we wanted to do now is to put, as I said, this on-off switch on the chromosome of the vibrio regions host and have it under some regulation of external signal. For proof of concept, we chose arabinose as the regulator. So when we add arabinose, the small gene here should be induced, and it should activate the type 6 secretion system from the plasmid to kill the rival bacteria. And we call this engineered vibrio regions VNAT-reg because it can regulate an inducible type 6 system. And this actually worked. If we look here, when we add arabinose in the blue circles, a vibrio regions attacker that contains this inducible type 6 system on the plasmid was able to kill our prey bacteria. Look at the drop in viability in the blue circles compared to a system to an empty vector. But if we didn't add arabinose, so the on-off switch was off still, the prey bacteria were growing, which we can see here in the green triangles. So basically now we have, in a safe bacterium, an antibacterial system that we can control and is responsive to the environment. Now I mentioned that the third thing we wanted to do is to customize the effector repertoire that it delivers. So every tail tube that is being shot delivers more than one effector. It delivers a whole repertoire of effectors with diverse activities. So can we actually control what they are, what these effectors will do? So to test that, we first had to get rid of the endogenous effectors that are encoded within the cluster that we cloned. And there are two regions with antibacterial effectors, shown here in red and green, and here at the end. So first we got rid of those to make what we call the effectorless type 6 system. So it's without the effectors and also without the regulator, so it's off. But when we add arabinose, it should be on. Even though it's on, this effectorless system cannot kill the prey strain. The prey strain grows. So we got rid of the effectors, but we know the system is still active and functional because we can actually see it being assembled inside the bacteria. What you see here are bacteria, and every line you see within them is a type 6 secretion system assembled. So we fuse a fluorescent protein to an outer sheath protein, and you can see the whole thing being made. You can see how huge these structures are. They're encompassing the entire width of the bacterium. So we have a functional system. It's not killing anything because there are no effectors. Can we introduce effectors from other bacteria into our system on a second plasmid and have our system work with that and kill other bacteria? To test that, we used 3 different effectors from 3 different other bacteria that we had. TME in green is a pore-forming toxin. PONE in purple is a DNase, and the last one in cyan is a phospholipase. We put each of them on a plasmid, introduced it into our inducible system, and now we do the same competition assays I showed you before, but now I'm showing you a different analysis of these assays. Instead of showing you the colony-forming units of the prey, I'm showing you some manipulation of that, which we call an effector toxicity index. Basically, it means how much an effector is toxic towards a prey strain. The higher the index is, the more toxic the effector is. And anything above the red line means toxicity. So you can see that all three of these effectors were used by our system to mediate killing of a human pathogen prey called Vibrio vulnificus. So we can control the effector repertoire of this inducible system. The interesting thing was that when we tested these three effectors against other pathogens, such as Aeromonas jandii or E. coli, we noticed that each effector, even though they target very conserved components, DNA, membranes, they actually have a toxicity range. So if you look at the pore-forming TME in green, it kills Vibrio vulnificus very nicely, has some killing ability against Aeromonas, but does nothing to E. coli. Whereas the DNase in purple kills all of them to different degrees. So we also learned something about the basic science of the system that we're now pursuing, which is to understand how something that targets DNA has a preference towards what is going to be the toxicity range. I'm not going to show it, but we also were able to see that if we introduce two effectors into the same bacterium, they can both be delivered and increase the toxicity range of the system. So what I showed you is that we actually have engineered what we think is a foundation for an antibacterial biotreatment. It's found in the safe bacterium, it's inducible and responsive to the environment, it's customizable, we can change different aspects of the system, most importantly, the effector repertoire to control the toxicity range. And again, I didn't show it, but the system is active under a really wide range of conditions. So we can see activation and killing of prey bacteria between 20 and 37 degrees Celsius, between 1 and 5% sodium in the media. And if you think about the properties of this system, the fact that each shot of this tail tube component delivers a repertoire of different effectors with different activities, and the fact that the actual delivery of the effectors is by brute force. There is no dependency on anything in the prey cell. There is no receptor that is required. You don't need anything from the translational or transcriptional machinery of the prey because we're delivering actual effectors, actual proteins. The combination of those makes the emergence of resistance unlikely. And so how do we actually get this to be something that we can implement? First, we need to change the induction. Obviously, arabinose is not going to be something we can use. What we want to do is change the regulation of the on-off switch from arabinose to something physiological, which will mean that we can sense the presence of the pathogen we want to kill. For example, pathogens make molecules such as quorum-sensing molecules. So if we can sense those and make the system active based on sensing these molecules, we can have a system that's only active in the presence of the pathogen we want to kill. We also want to work on specificity. So as I said, as I showed you, we have toxicity range for each effector, but it's still too wide for us to not have dysbiosis or target undesired bacteria. So we plan to engineer strain-specific toxins that will really kill only specific bacterial strains. And of course, this is a genetically modified organism. We need to bypass government regulations. So we'll have to implement a few safety measures, such as kill switches that will make sure these bacteria don't just run off to the environment. So I'll finish by thanking the people doing the work. This work was led by Biswanath in the lab with the aid of Kinga. I want to thank our funding agencies, especially the ERC that supported most of this project. If you want to learn more about this project or other things we do in the lab, visit our website or follow us on Twitter. And thank you for the attention. I'll be happy to answer any questions.
Why would the bacteria find it stored in adjacent bacteria if they belong to the same culture and cannot identify itself?
So it kills non-keen bacteria because each antibacterial toxin that I showed you actually is encoded together with an antidote, an immunity protein that prevents it. So your keen have the antidote, rival bacteria don't. So that's what, currently, yes, it would, because it's indiscriminatory. Even with changing or controlling the effector repertoire, as I said, it's still too wide of a range. But I hope we have some ideas on how to engineer strain-specific effectors and then introduce those into the system and have something more tailored. I really hope so. Ask the governments. Yeah, there are no mice. This specific bacterium doesn't do well in mice. Again, it's a proof of concept. Actually, we're thinking about this bacterium more towards aquaculture environment because it's a marine bacterium and it's more suitable for that. The problem with that is that arabinose doesn't work very well with marine animals in the gut. I don't know why. So we'll need to change the regulation there to have something that's going to be functional. Thank you very much.